Tony, please.
Thanks, Sassy. I'm happy to take questions as we go through. If I confuse anyone, please stop me. And thank you very much for that great introduction. And what I hope to do in the next little while is show you some assays that will allow us to interpret the phenotypic implications of some of that sequence data. And I want to tell you about an assay that we've taken a lot of time to develop that gives us the sensitivity and high throughput that sort of complements the extreme power of the high throughput sequencing that you've just seen. So that not only can we look at the mutations, but then look at the phenotypic implications of those mutations, both in terms of immune avoidance, but also in terms of tropism, because tropism determines how severe the infection is. Clearly, the early virus was more pathogenic than the more recent strains of the virus, and that seems to be associated with altered tropism, which relates to the receptors to which the spike protein binds to. So these are my conflicts of interest, and none of which really impact on anything I'm presenting here, but some of those clinical trials have been of a hyperimmune globulin, and some of my interest in these assays was really around identifying effective neutralization, people with effective neutralization titers, so that we could build good hyperimmune globulins to give to people. So in this talk, I wanted to talk about the development of some of these modular assays that allow us to characterize the virus in terms of its ability to interact with the immune system and the effectiveness of neutralizing antibodies against different variants as they arise, and to describe the viral evasive capacities. And this, what I want to tell you about is how we developed really susceptible human cell lines and a way of building a modular virus so we can try and keep up with the variation in the virus so that we can keep producing these results. As my colleague said, some of these viruses are very, very difficult to grow. And what we've tried to do is develop a system that is malleable so that we can adjust the system to isolate the virus no matter how difficult it is to grow. And then I want to show you some data at the end that shows you that in someone with in all of us after infection, our immune system doesn't rest, and there's actually active evolution of the neutralizing response over time. That happens in individuals, but it also happens across populations. And I want to show you some data that demonstrates that, which shows the power of a polyclonal response over the monoclonal response. As we know, all our monoclonal antibodies that we developed are now therapeutically useless because of the viral evasion. But these polyclonal responses seem to maintain efficacy against the virus. So how do we measure antibodies against SARS-CoV-2? Well, there's a range of ways of doing this, and the easiest way of doing this, and the assays that became rapidly available were ELISA-like assays. That is, we have an antigen affixed to a solid media, and then we put serum into that, against that solid media, and the antibody binds, and then either directly or indirectly we have a conjugate that makes a colorimetric change, and and the deepness of the color tells us something about the concentration of the antibody that has been produced to the antigen. So this is standard technology. These were developed very, very quickly and became commercially available pretty early in the epidemic. They're very, very useful as diagnostics. They're easy to standardize and to automate. We know a lot about them, and they can, and in certain circumstances, you can quantify the immune response using a standard curve. But they may or may not correlate with useful viral immunogenicity. They tend to measure antibodies that bind to that antigen. They may not alter the efficacy of that antigen. So the most important antigen on SARS-CoV-2 is the spike antigen, and you can have antibodies that bind to all parts of that complex structure that you were just shown, but it's only the antibodies that bind to certain parts of that structure that prevent the virus entering the cell, because it disables the function of spike, that are important, and the ELISAs don't usually measure that. So how do you get around that? Well, you can develop these pseudovirus assays. And so this is now a functional assay. They're relatively safe because what you're doing is you're putting the spike protein onto a backbone of another virus, like an inactivated lentivirus. And you're looking at the ability of that virus particle to fuse with and get into your target cell. So these are visualized by either colorimetric or fluorescence change inside the cell, and the readouts are either direct microscopy, fluorescent microscopy, flow cytometry, or fluorimetry. And these measure antibodies that inhibit that first entry step in the virus life cycle, and therefore are measuring a functional consequence of those antibodies. They're much more sophisticated, they're much harder to standardize and to automate. They generally correlate well with functional neutralization, unless there are complex permutations of viral interactions with the cell receptors. And what we've learned about SARS-CoV-2 is that there are complex interactions with both ACE2 and a protease, TMPRSS2, and how those interactions occur with the different variants actually have changed over time. And that is actually what determines the tropism of the virus and whether it targets the upper or lower respiratory tree. And also the interaction of those proteins on the membrane with the nuclear capsid is very important in terms of the viral pathogenicity and its ability to replicate. And so if you don't have that viral structure, that true viral structure, but you have a retroviral structure supporting those membrane proteins of the virus, they may not faithfully reproduce what's actually happening with the virus. But these are pretty good assays, and the other advantage is that these are relatively easy to set up in if someone understands and is good at genetic engineering, and they can be done in a PC2 lab. They're safe, your staff aren't going to get infected, they don't have to grow high titer virus, but they may not always replicate what's happening in vivo. We have spent a lot of time, so we've developed some of those assays, and I'm not going to talk about them too much here, but we've spent a lot of time developing these live virus assays. So the big disadvantage of live virus assays is that they are live virus assays. They're with pathogenic virus, and so they have to be done in a PC3 lab with full PPE. And so they're not something that can be used in a standard diagnostic assay, but they do give you very important information. And what I hope to show you is that they're highly adaptable to this highly adaptable, immutable virus that has evolved at very rapid rates over the last 3 to 4 years. So these are functional assays. The readout is either inhibition of viral entry or actually inhibition of cell death. So the virus gets in, it kills the cells. If there's something that inhibits the virus getting in, the cells won't die. You've neutralized the pathogenicity of the virus. In our hands, this is the most realistic in vivo replication of what's actually happening in vivo. It's replication of competent virus, but that means that the virus has to be isolated and then titrated and used at particular concentrations in these assays. So a standardized amount of virus has to be added to the assay. As I said, it's visualized by cell death. This is often called plaque formation. If you're directly visualizing that by looking down a microscope and counting the amount of cell death, but you can substitute this, as we have, with colorimetric or fluorescence change that detects the cell death by counting cell nuclei. And so the readout is usually microscopic, which if it's done by a person, it's highly labor intensive, it needs great expertise, but this can be automated with high content microscopy and fluorescent dyes that stain nuclei. These measure antibodies that inhibit infection and also can be used to measure the pathogenicity of the virus. These are far more sophisticated and complex. They're much harder to standardize and automate. And as I said, they require a PC3 lab and highly experienced staff, but you can learn a huge amount from them. And you can standardize these assays against the WHO standard, and you can automate these by having automated microscopic platforms that count the wells in 384 well plates. So you can change these things into really high throughput assays. So the standard live virus assay is using the Vero E6 cell, which is a macaque kidney, sorry, it's not, it's an African green monkey kidney cell line. And these, as I said, depend on a reader looking down, a person looking down a microscope and quantitating cell death against titrated dilutions of a serum. It's very label intensive. And the other issue, and I was very sensitive about this, I have to say, is that this is not a human cell line. This is an African green monkey cell line. I'm a pretty old man now, but we lost 10 years at least in HIV research, and particularly around vaccine research, by studying lab adaptive HIV that were grown, those isolates were grown in monkey cells. And they adapted to those monkey cells, and the results from those assays meant nothing in the end in terms of vaccine development. In fact, they pushed us down wrong pathways. So I wanted to get away from Vero E6s as rapidly as possible. And fortunately, the guy that set these assays up, Stu Turble, and I thought very similarly on this, this issue. So we rapidly started thinking about human cells for how we might do this. So what we've developed is a suite of closely related assays which we can take a new sequence and a new isolate, and within a week we can tell you what the neutralization profile of that virus is, how immunoevasive it is, and we can have a guess at its tropism and its replicative ability with this suite of assays. These allow, and so we have a human cell line which is based on human embryonic kidney cells that have been transfected with ACE2 and TMPRSS2 and then screened for permissiveness. And we actually have a library of these cells with different levels of ACE2 and TMPRSS2 expression. This allows both rapid virus isolation and then phenotyping of each variant of concern. And we can isolate these even when the CT values on the swabs are very high. And so this allows us rapid characterization of the virus. And combined with high throughput sequencing, this allows rapid genotype-phenotype correlations. And we can then, because it's a high throughput assay, apply this to appropriate cohorts and samples. So just to harp on about this a little bit more, there are two ways that the virus gets into cells. One is this endocytic pathway, which depends on it binding to cathepsin. This is the pathway that occurs in Vero E6 cells, and this is how the virus gets in. In vivo, we believe this is a minor pathway. The main pathway that, by which the virus gets in is by spike binding to ACE2 and then being and the spike being processed by TMPRSS2, which is a protease, and then entering at the membrane, which we believe is the major way of getting the virus getting into cells in vivo. And the viral tropism is determined by the viral spike's relative dependence on ACE2 and TMPRSS2. And so we decided to build an assay that allowed characterization a cell line using different levels of ACE2 and TMPRSS2. So I think I've been through most of this. I've just said most of that. And this is not to say that Vero E6, so Vero E6 is still sort of the gold standard and the standard used by most laboratories, and so is a very effective assay. But I think it has allowed us to really, this assay has really allowed us to rapidly look at each variant of concern as it's arisen. Actually, the real gold standard is to do all this in cells that the virus really infects, which are airway cells, and doing primary culture of those airway cells, either the bronchial cells or the alveolar cells. If the assays I've described to you are difficult, these are far more difficult. It takes weeks to get those cells to grow up and differentiate in culture. You have to have a real green thumb at tissue culture doing that, and just growing up the cells for targeting for your assay takes between 4 to 6 weeks. So this is not something that can be adapted to a high throughput assay, but is the gold standard. And if I get time at the end, I'll show you where we've actually compared our assay with the viral characteristics in cells derived from bronchial epithelium. But if you need to validate the assay against both those and the way the virus behaves in vivo. So we chose to take this modular approach because what we learned very early on, and as the first talk demonstrated, this virus has evolved very, very rapidly. And so we've taken two approaches to being able to characterize this virus. One, we've built a modular SARS-CoV-2 backbone that is sort of the equivalent to PNL 4.3, which is a plasmid form of, full-length plasmid form of HIV that we can cassette into and out of the various spikes. And we use that for our pseudovirus assay, but we also have used that for reverse genetics building of SARS-CoV-2 for introduction into the live virus assay. But we've also built, so we've played with the virus, but we've also built these hyperpermissive cell lines that are human cell lines that are 10 to the third to 10 to the fourth to the fifth, in fact, times more sensitive in terms of allowing viral growth. And this has been able to accommodate the various strains of the virus over time. So this is the basis of the assay. Basically, you get these cells, whether they're Vero E6s or these hex cells that are transfected, to go to confluence in wells. This is a 384-well plate here. We do it in a 384-well plate, and then you infect each well with the virus, with a known quantity of virus, with a pre-treated virus, and then you quantitate the cell death by looking basically at either the plaques, which are these empty spaces, or the way we do it is by counting live nuclei in live cells. And by counting the nuclei in live cells using a live dead dye, this allows us to automate it. So these 384-well plates are fed into a high content, high throughput microscope, and each of those wells is assessed for the number of nuclei in a predetermined field of each of those wells is counted, and then that gives us the amount of cell death in each well. And that's obviously done in replicates, and then that allows us to look at the extent of neutralization in someone who's generated an immune response after vaccine, or in a negative control shown here. And we've really, a lot of this has been learned from assays that we had tried to develop for similar approaches to HIV. And so it's miniaturizing the assay into 3 84-well plates and then having a high-content microscope in the PC3 lab that allows you to assess these plates very, very rapidly and having a binary readout. So there's either nuclei there or there's no nuclei there, and then counting those nuclei. And we cross-validated this against reader-scored plaque assays in over 3,000 samples. And this is just demonstrating that these are these cells. Again, these are the transfected HEC293 T cells, and these are the HEC293 T cells untransfected. And then using, in fact, in this case, a pseudotype virus, looking at whether you actually get fusion of the fluorescent virus into the cells. And you can see that either with the delta with the spike from delta on the surface of these pseudovirus particles, or pseudotype with VSVG, you get good infection of the HEC239s. But because HEC293s don't have ACE2 or TMPRSS2, you only get the fusion when you have VSVG on the surface of the pseudovirus particles. So again, this is just a blown up of a negative control well. The cells are confluent. You can see they're in a confluent single layer across the floor of a 384 well plate. And these are as a well with considerable cytopathic effects. And so you can count the relative cell death and quantify that in replicates, and you can add various levels of serum or monoclonal antibodies to these wells and titrate out and get your sigmoid curve here in terms of your neutralization. So you can see that in this particular sample, the IC50 or the concentrate, the titer at which you get 50% neutralization is 1 in 640 here. So this is just showing how the plasmid that we've developed that allows us to chop in and chop out the various spike proteins. And we can slot in the various spikes from the various variants. These are all the early variants, and get fusion and get a readout. So the other challenge that we tried to address is that essentially we were getting samples from quarantine. We would get one chance at that sample. There would be one swab taken, and if they came back with, if it came back as positive but had a low CT value, we would be unable to isolate the virus. And in general, with VIRO-E6s, you get about, from swabs collected like that, you get about a 10% rate of being able to grow the virus with the relatively easy to grow viruses. And certainly you need, usually need a CT value of less than 20 to get growth in VIRO-E6s. So we looked, the other reason for using these transfected human cells was that we tried to really screen a library of these for truly hyperpermissive cells, so that we could then take swabs that people could still get sequence from and grow the virus from those swabs, so that we could do the genotype, phenotype characterization, and we could do that efficiently, so that when we first saw, we had a good chance when we first saw the new sequence of actually being able to grow that virus up and characterize it. And so, again, just to remind you that the main mode of entry is through the membrane. And so this is just some graphs of the viral dilution you require to get growth in various cell lines, and you can see that you need a lot of virus to get a viral readout in Vero E6s. With our with our best or our standard HEC clone, which is clone 24, we can use essentially swabs with 10 to 100,000 times less virus and still get a virus isolation compared to the Vero E6s. And this is just showing that we don't always get isolation, but we can get isolation out to CTs of 35, which is quite low, and certainly at the limit of where you'd get a sequence of that swab. This is a very hyperpermissive cell line. And when you get this assay humming, you can get these very nice sigmoid curves that have a very nice R value. And these, you can see that the error bars, well, you can't see the error bars because a lot of times the error bars are very, very small. You can see some error bars here, but this is a very reproducible assay. And you can see that whereas these are the early clades, and this assay doesn't give you a lot of advantage when isolating clade A or clade B. As the virus has evolved, it gives us more and more advantage. So the Vero E6s are shown in the clear bars here, and our HEC clone 24s are shown here, either after 72 hours or in fact 24 hours here. And you can see that there's not a huge advantage with these early clades, but with the later clades, you get much higher titers of virus, much higher levels of isolation than the Vero E6s. So that's what a standard assay looks like. So these still take a lot of work to set up. And then what we can do is, this is an early experiment where we mapped a response to a Pfizer vaccine over time at 1 week, 2 weeks, 3 weeks post the first vaccination, and then 1 and 2 weeks after the fourth vaccination. And you can see the increasing IC50s here from undetectable up to an IC50 of 209 here. So it clearly gives us a dynamic range. We could demonstrate the efficacy or otherwise of various monoclonal antibodies, and we could look at vaccine responses against a range here of early variants, and you can see that there were quite some differences here in the susceptibility of the various strains to the neutralization antibodies that were developed by Pfizer, by the Pfizer vaccine. So what I wanted to start talking about was some of this data here, where we started looking at hyperimmune globulins. So we were trying to make pooled serum from, pooled IgG from people who had either been infected or immunized. And we wanted to identify people who had high neutralization capacity and then pool those together to perhaps produce a polyclonal therapeutic. This is a batch of hyperimmune globulin that we made in Australia with a company called CSL, and the IC50 against a range of the early strains. And you can see against the early strains, beta was by far the most immunoevasive. It it took the most hyperimmune globulin to neutralize. Whereas Delta really wasn't very immunoevasive at all compared to the Alpha strain. Beta was far more immunoevasive. Whether you looked at post-vaccine donors or people who were during convalescent after natural infection. As we know, Omicron had lots and lots of variants, as was just beautifully shown, and it became a soup of Omicron variants that everybody was exposed to. And just looking here, perhaps concentrating on this, again, hyperimmune globulin batch, this is the same graph now showing how much more immunoevasive Omicron was than the early strains, including Beta. So it was, if this is how much hyperimmune globulin you needed to neutralize Alpha, you required 10 to 15 times as much to neutralize Omicron. And again, this is whether the hyperimmune globulin came from convalescent or from people who were convalescent and who had been vaccinated. You still had this 10 to 15-fold increase in the amount of this hyperimmune globulin that you required to neutralize the virus. So we continued this collaboration with CSL, but in this case, CSL in the US. And what they were doing is they're making Privigen, which is intravenous immunoglobulin, which is pure pooled IgG that you give to people who are immunodeficient, who can't make their own antibodies. So it's a therapeutic. We use it as immune replacement, and in some people we use it to treat their autoimmune diseases. CSL make a batch of this in the US from 10,000 donors every 3 weeks. And so they get 10,000 donors, they make a batch. Another 10,000 donors make a batch, and that is every 3 weeks. And they would send us a batch of this intravenous immunoglobulin every 3 weeks for testing. And so we tested their immunoglobulin, and so we could look at a snapshot of the way the immune response was evolving in the US over time. And because we had this assay and we had all these variants isolated, we could look at how this hyperimmune globulin evolved over time, but how it also neutralized various of these strains, including the Alpha strain, Delta, and then all these variants of Omicron here that I'm not going to go through. But this is the time period here. This is when Delta was circulating. This is when Omicron BA1 and BA2 were circulating. The black line is the response in the hyperimmune globulin, the neutralization titer in the hyperimmune globulin to the Alpha strain, and this is to Delta. And as I said, Delta didn't look very immunoevasive at all. And you can see that the immunity in the American population, as judged by this, increased rapidly during this period post. These are the numbers of vaccines given at this time. And this is one of the waves, the Omicron wave. This is the number of infections through the Omicron wave. So you can see that the titers of antibodies in these batches of intravenous immunoglobulin followed increased following the increase in vaccination in the US. So this allows us to follow what's happening over time. What I think is really interesting is that there were increases. So this is the BA1 and BA2 are shown in red and blue here. This is a measure called breadth, and this gives us a little bit more dynamic range. And it again reflects neutralization titer. And what you can see, I think, which is really interesting, so it's a relative neutralization relative to the alpha strain. And you can see that there was already increasing neutralization capacity to beta, to BA1, before BA1 was circulating in the population. So again, the polyclonal response, and this increased over time as people were exposed to BA1. But again, before BA2 was in the population, there was increasing immune responses to BA2 prior to, and then once BA2 became detectable, there was a rapid increase in responses to BA2. So within that polyclonal response that we're all making, there is the capacity to neutralize variants that we haven't even seen yet. So we've continued to do this, and this is more recent. This is out to January 23 with some of the more recent variants shown here. And again, you can see the same trend. And the other thing I think to note is that despite all this changes and resorting of mutations in Omicron, there's only about a 5-fold difference in immunoevasive capacity between all these more recent strains here. And so it's almost like the virus is boxed and it's trying to find a way to become more immunoevasive, but is only able to change its immunoevasive capacity within a relatively low dynamic range. And this, I might just skip that. But we can use this to look at the efficacy of both standard vaccination and bivalent vaccination, which is what we've shown here, and against each of the strains. And then, you know, the most recent worrying strain is JN1. And this is now data again from Australia. And this is the neutralization capacity against various Omicron variants. And you can see that JN1 is no more immunoevasive than a lot of these earlier strains like XBF or XBB1, XBB1.1.5. And this is shown here. This is with Australian pooled donors, and this is with US pooled donors, so very similar. I'm not going to go through all the background to these assays, but by tweaking these assays, we can get an idea of the replicative capacity of each of the strains by comparing what we get on the diagnostic swab against the viral titer. So it's the ability of viral particles to produce viral titer. And you can see that of the strains, the strain that was tenfold better than any other strain at producing virus per CT value was Delta. So it produced more live virus per virus particle infecting a cell than any other of the variants. And I don't have the data here, but that includes JN1. So Delta was the champion at producing new viral particles from a single viral particle. This is looking at tropism here, and this is now looking at bronchial epithelial cells, and you can see that Delta was pretty poor at infecting bronchial epithelial cells, whereas BA2 and BA5 were much better. And they established infection much earlier, and it took Delta much longer, 6 days, to establish high levels of virus infection in bronchial epithelial cells. But Delta was much better at replicating and much faster replicating in alveolar cells. So consistent then with what we saw clinically, that Delta was much more pathogenic than Omicron. And so Omicron seems to be targeted in the upper respiratory tree. This is, and we think we can get a handle on this. We're looking at various algorithms to sort of convert this observation in primary tissue culture cells with the degree of TMPRSS use by blocking entry, how much use of nafamostat, which blocks TMPRSS function, how much that attenuates the ability of the virus to get into our cells. And then data that I won't show you is that we have, again, a library of cells with different levels of ACE2 and TMPRSS2, which we think we can use to mimic these readouts here. But the virus replicate with this, as I said, these cells take about 6 weeks to grow up, and then the assay takes 6 days to do. With our cells, there is cell death already at 6 days, 6 hours, and so we can get a readout at 20 hours rather than 6 days. And then just again, I don't want to go through all this because it's very complex, but again, using these cell lines, we can get an idea of tropism, as I said, and you can see that JN1 here, these are all the other, so this is alpha, beta, delta, and then a bunch of Omicron variants up to JN1. And JN1 behaves very, very differently in this assay, and this is sort of the fold enhancement of infection with ACE, when ACE2 is cleaved by TMPRSS2. And you can see that JN1 is a real outlier. So it seems to be very, very ACE2 dependent and not TMPRSS2 dependent. So although it's very infectious, we don't think it's going to be very pathogenic. That's our prediction. So this is a summary slide showing that we can take clinical cohorts, we can take the sequence from those clinical cohorts, and take clinical samples, primary viral material from swabs, and plug those into these high throughput assays that generate a range of rapid characterization of the virus, including its immunoevasive properties, and then correlate that with the whole genome sequencing that our clever colleagues who do the sequencing do. So I'll leave it there. Thanks very much.